Thank you for the organizers for the invitation, and I'm very glad to be here to talk about the hamsters and how this small animal can teach us a lot of COVID and long COVID. So I have nothing to disclose. Back in the beginning of the pandemic, in 2020, we started to study the mechanism of anosmia in human patients, and we detect that people having presenting a loss of olfaction have olfactory sensory neurons infected in the olfactory mucosa. You can see here this yellow cell. And we try to transpose this feature in the hamster model by infecting them. And we discovered that the infection promoted a complete destruction of the olfactory mucosa in the hamsters, with the infection of olfactory sensory neurons and also infection of fibers going from the nasal cavity to the olfactory bulb. And looking deeper in the olfactory mucosa, we detect that there was a massive deciliation in the olfactory mucosa. And in this area, the cilia, they are part of the olfactory sensory neurons. So we focused then to try to prove that the SARS-CoV-2 could infect the neurons and then reach the brain using the hamsters. We started to work with the U1, the ancestral virus, but then Gamma, Delta, Omicron, BA1 arrived, and we have these 4 viruses in the hamsters. And what is interesting to see is that the animals presented the same clinical differences manifested by people. So the U1 was the most severe virus, and then Gamma, Delta, and Omicron being the less severe. And in this graph, we put the olfactory performance of the animals. We tested the animals to see if the infection would cause a loss of olfaction. And here, the U1, so it caused lots of 62% of animals loss of olfaction. Gamma, there was no anosmia, but we can talk about hyposmia. You can see the performance is delayed, but they find the food. This test is based on the buried food finding test. We hide food in the bedding, and then we measure the time that the animals take to find the food, as having only the olfaction as output. And here you can see also the lung weight. So all the animals presented signs of pneumonia. There was exudation, there was inflammation and hemorrhages. But we were more interested in the brain. So we could take the brain tissue of the animals, and we found that the olfactory bulbs were infected. All viruses were detected in the olfactory bulbs, regardless of the presence or not of olfaction loss. The virus was in the olfactory bulb of this animal. And the olfactory bulbs, there was also inflammation and antiviral response in this tissue. But we could isolate the virus in the olfactory bulbs, but then we'd like to see the infected cells in the brain. So we went to the Paris Brain Institute and we performed a light sheet imaging in the head of the animals. You can see the orange cells are infected cells in the nasal cavity. And you can see there is virus everywhere. And going to the olfactory bulbs, you can also see some infected cells. It's more rare. We can also see infected fibers. And the virus is there. It's the viral load is low, but we could prove that the virus reached the central nervous system via the olfactory bulbs. And later, we want to prove again that the virus could indeed reach the brain via the olfactory bulbs, and reconstruct recombinant viruses expressing nanoluciferase. Nanoluciferase is an enzyme that produces light. And then we infected the animals with U1 nanoluciferase and delta nanoluciferase. And again, regardless the absence, the difference of the clinical manifestations, we could clearly see Here, the olfactory bulbs showing light, emitting light, and this is indicative that the virus is there and the virus is replicative. We published these results recently, and the message of this paper was all the variants can invade the brain via the olfactory bulbs, regardless the difference of the clinical manifestations. And you also put the message, but I won't talk here, but the virus can infect the neurons and can move inside the axons. And the virus can move in the anterograde and the retrograde directions inside the axon. So, coming back to the long COVID, at the first, our first study, we stopped to study the hamster at day 13, so 2 weeks, and we detect that the acute phase was around 5 days post-infection. You can see there was blood weight loss, the clinical score, and the olfactory depth was finished by day 13. But later on, we thought that day 14 perhaps was not enough, was not long enough to see the long-term effects for long COVID. So we came back with the U1 Delta Omicron BA.1, and we infected the animals and keep them during 80 days. It's very long for a lab experiment, and you can see here we have the acute phase for all the animals, and then around day 15, day 30, and day 80, we tested these animals for different outputs. We were interested in the anxiety, depression, and memory loss. And what is interesting with this model is the somatic symptom disorder free. They don't think they are sick. So it helps us to be objective in the output. So the first test was the anxiety, the test for anxiety, and it's a test to measure conflict. So we put the animals in a new arena with food inside in the middle, and we measure. So the animal wants to eat, but it's an anxiogenious environment. So we will measure the time that the animal go there and take the food, and you can see. We can see in these graphs that we have for U1 and Delta in Omicron infected animals, we have this tendency to pass more time before going there and taking the food. So it's indicative of anxiety-like behavior for these animals. The second was for depression, and for depression we measure anhedonia, so it's the ability of the animals to feel pleasure, and how we measure that, we spray the animal with sucrose, it's very viscous solution, and then we measure how long they spend grooming themselves as an index of self-care. And this output was more easy to see in female. In female, you can see that the U1 infected females have all the time below the mock-infected animals. And Delta and Omicron, it was like this relapse same symptoms. We have depression-like behavior at day 13. At day 30, the animals they seemed healthy, but at day 80, they were presenting again a depressive-like behavior. And the last test was for memory, how we measure memory in animals. So we put the animals with 2 objects. They will recognize these 2 objects. And then in a second session, we change one of the objects, and we measure how long they will explore the new one. Because they know that the old one is a non-object, so they keep and pass more time exploring the new object. And you can see that all infected animals passed less time exploring the new object. So there was an issue with the memory of these animals. And then I decided to put this painting to make like a cultural interlude so we can think about this. And it's a painting from Goya. It's just there in the Museo del Prado. And it's very interesting because this painting calls the dog. But if you don't know there's the dog there, we don't know what to look for. So I think it's very nice to put it in the long COVID. We need to know who, where, and how look for the viruses. We also took the brainstem of the animals and performed bulk RNA-seq. For those who are not so familiar with the technique, so we take the brain and we sequence all the messenger RNAs in the tissue, And then we will find the genes that are up or down regulated and try to find common pathways with these genes. And if you see these terms here, there are some terms that are interesting, like Parkinson's disease, Alzheimer's disease. I'm not saying that these animals had Parkinson or Alzheimer, but the genes were related with this disease. And if you go to the gene ontology terms, to see what biological processes were dysregulated, we can see mitochondria, cytochrome, proteasome. And those who work with neurodegenerative disease know these terms, I think, very well, better than myself. And going for the down-regulated pathways, the list is even bigger. So I'll focalize just in few ones. I have here downregulation in the glutamatergic synapse, in the cholinergic synapse, in dopaminergic synapse. I think all of you have listened to the glutamatergic, cholinergic, and these neurotransmitters. And just to sum up all of this information with the RNA-seq, there's lots of information, so we need to focalize in one or another. I took these two images to try to find common pathways with pathological aging. And again, the hamster, they are not old, and they don't have any neurodegenerative disease. But if we go one by one, we have lots of proteostasis in the brain of the animals. The mitochondria are not working very well. The stem cells are exhausted, and I want to focus on that because the neurogenesis is very impacted in the brain stem of the animals. And the communication between cells are quite dysregulated, especially for neurons. The communication is related to neurotransmission. And again, in age-related neurodegenerative disorders, we have diminution of the glutamatergic system, the cholinergic system, and the dopaminergic system. And all of this data we are still validating, and we want to validate what we find in the hamster brain. We want to validate in human cells to make the bridge between the animal model and human model. And here you have human neurons differentiated from stem cells, co-cultured with viral permissible cells. So what you see in green is infected cells, what you see in orange is neurons, and in blue is the nuclei. And doing this kind of culture, we can see infected neurons, and we can validate what we see in the RNA-seq from the hamster brain, including the diminution of neurogenesis in a dish. And the take-home message that I wanted to say here is that I hope I convinced you that SARS-CoV-2 is neuroinvasive, and SARS-CoV-2 persists in the brainstem, at least day 80. And I put a diagram before, so 80 days in a hamster lifespan is not 80 days at the human lifespan. We need also to consider time in the species. And the SARS-CoV-2 induces a long-term neuropsychiatric and cognitive sequelae in the hamster model. And finally, the long-term SARS-CoV infection may reduce neurogenesis, which I think is an important point, and one point that I'm going deeper to try to understand. The long-term infection may impact brain connectivity, especially if some specific neurotransmitters like glutamate, dopamine, and acetylcholine. And finally, it may mimic neurodegenerative disorders. And this is one point, at least for the neuro part of long COVID, that we need to focalize to try to understand how this virus is inducing symptoms and manifestations related to neurodegenerative disease in non-old brains. And to finalize, I'd like to thank the founders, all the collaborators, and you for listening to me. Thank you.